Today we're going to talk about the citric acid cycle, which is a metabolic pathway in your body. It takes place in the mitochondria of eukaryotes and in the cell body of prokaryotes. So let's go ahead and get started. In step one, as you can see, it's a condensation, and you have your acetyl-CoA, and it's acted upon by citrate synthase, and a water comes in, and a CoA-SH comes out. And what you're left with is citrate. And if you'll notice, citrate is a 6-carbon molecule, and that's very important. For step 2A, you have your citrate. And let me just say, I've color-coded these carbons to so that you can follow along with where the carbons begin and where they end up in the entire citric acid process. So you start with your citrate, and you go, and it's a dehydration reaction, so water is going to come out of it, and you're left with cis acetate, and all you're losing is that alcohol. And then next is step 2B, and this is a hydration reaction. And so you have your cis acetate, and water is going to come in, and it's going to put that alcohol on the lowest backbone carbon by the third carboxylic group, and that's going to give you isocitrate. Next, for step 3, you're going to start with isocitrate, and it's an oxidative decarboxylation. So what this means is it's an oxidation reaction, and you're losing a carboxyl group. So you start with your isocitrate, and isocitrate dehydrogenase, which is an enzyme, works on it, and NADH is your first cofactor, and then you're left with that CO2 and an NADH and then an extra hydrogen. And it takes out that middle carboxylic group, and then you're left with alpha ketoglutarate. For step 4, you start with alpha ketoglutarate, and you're going to have another oxidative decarboxylation. And what that means is your first two cofactors are NAD plus and CoA-SH, and alpha ketoglutarate dehydrogenase is going to work upon the molecule, and you're going to end up with CO2, NADH, an extra proton, and then succinyl-CoA. And so you've lost 2 of your carboxylic groups, and so now you've gone from a 6-carbon molecule to a 5-carbon molecule. Now you're at a 4-carbon molecule. For step 5, it's a substrate level phosphorylation, and so you start with your succinyl-CoA, and then your first cofactor is GDP and a phosphate, and your ending group is a GTP and a CoA-SH, and succinyl-CoA synthetase is your enzyme, and you're going to end up with succinate. And I've stopped using the color coding, but because at this point the molecules are somewhat symmetrical, and so that CH2 and that carboxylic group can be either of those 2 groups. You really don't know. And then for step 6, it's a dehydrogenation, which means you're going to lose 2 hydrogens. So you start with your succinate, and your first cofactor is an FAD. It also can be annotated as a Q. And then your end cofactor is a FADH2, and your enzyme is succinic dehydrogenase. And that creates a double bond between the 2 central CH2s, and you're left with fumarate. And then for step 7, it's a hydration, so water is going to be acting in this reaction. And you start with your fumarate, water comes in and works with fumarase, and you're left with malate. And so it destroys that double bond, it goes back to a single bond, and it adds that alcohol. For step 8, which is your final step, it's a dehydrogenation. And so you start with your malate, and your beginning cofactor is NAD+, and your ending cofactor is NADH and another proton, and your enzyme is malate dehydrogenase. Should have mentioned this earlier, but enzymes are all in purple and cofactors are all in red. And your final product is oxaloacetate, which is an incredibly important molecule. And the oxaloacetate can be used to start the citric acid cycle again. So it's almost a catalyst for itself. And it can go off and do other things. And I think in biology and chemistry it's important to look at the entire picture. So on this slide, I've drawn out the entire reaction, but instead of putting the structures, I've put the number of carbon in each molecule and made it representative so you can look at it. And you're starting with pyruvate, which you got from glycolysis, and then you're just going through your reaction and you're losing a carbon between isocitrate and alpha-ketoglutarate, and you're losing another carbon between alpha-ketoglutarate and succinyl CoA. And I've only put the ending cofactors like NADH and FADH2 and GDP, mainly because I didn't have a lot of room on this particular slide. And then this slide shows the various molecules involved in the citric acid cycle and what they go to make. And so if you want to think of the citric acid cycle in its largest form, what it essentially is it's a big chemical manufacturing process that takes place in your mitochondria. So think of it as a factory. So your citrate can go to making fatty acids and cholesterol. Alpha-ketoglutarate can go to make amino acids and nucleotides, which we all know is incredibly important. Hashtag biochem1. Succinyl CoA can go make heme, which is the central protein molecule in your hemoglobin, which carries oxygen through your body. Malate goes to make pyruvate, which begins this entire process. And then oxaloacetate goes to make glucose, which we all know how incredibly important that is, an energy source, yada yada yada. On this slide, I wrote out the net equation. So you have your acetyl-CoA plus GDP, plus PI, which is a phosphate molecule, plus 3 NAD pluses, and then plus an FAD plus. And your products are 2 CO2s, your CoA, a GDP, 3 NADHs, and an FADH2. And then a mnemonic that helps me remember this is you can pretend you're a poor college student and you're at your concert and you're selling shirts that you made yourself, and that's a trademark violation. And so the manager for the band calls the police to have you shut down, and the police come up to you and you just beg the police officer, "Can I keep selling shirts for money, officer?" And in mnemonics, the beginning letter stands for something. So your C in tan stands for citrate, your I stands for isocitrate, your K in keep stands for alpha-ketoglutarate, your S in selling stands for succinyl CoA, your S in shirts starts with succinate, your F in fumarate your F in four stands for fumarate. Your M in money starts for malate, and your O in officers starts for oxaloacetate. So, can I keep selling shirts for money, officer? And another thing people can get confused on is which goes first, succinyl CoA or succinate. So something you can use to remember that is succinate is always late, meaning it comes second after succinyl CoA. And then on this final slide, I talked about inhibitors and regulators of the citric acid cycle. And your inhibitors are citrate, which is one of your molecules, NADH, which is a cofactor, and succinyl CoA, which is another molecule. And so the inhibitions take place at step 1, and that's inhibited by all three, citrate, NADH, and succinyl CoA. Step 3 is inhibited by NADH, and step 4 is inhibited by NADH and succinyl CoA. And by inhibiting, I mean that they're inhibited when there's a large amount of these molecules. And then regulators for the citric acid cycle are calcium and ADP. So step 3 is activated by calcium and ADP, so it begins that citric acid cycle process. And then step 4 is also activated by both of them. And that, in a nutshell, is your citric acid cycle. And just remember to look at the entire picture, use your mnemonics, and just use flashcards. Thank you.